Okay, thank you. And thank you for the invitation to come here once again. I must say that I've been here since 2011, and this meeting has been the one by far with the highest scientific content. So it's very encouraging for the field. Really, good groups are working hard to help us understand this disease better. Disappointingly, there are very few clinical studies. We are the exception. But well, we're doing our best, and now I'll tell you about the Norwegian efforts in clinical trials. We have 2 clinical trials we'll give you some brief reports on here. It's a study, the RITUXME study is well known. It's headed from Haukeland University Hospital, but it's a cooperation between 5 hospitals in Norway, among them the 4 regional university hospitals and 1 community hospital. And I'll present the data on behalf of these researchers. We have to go back to where it started, namely a lymphoma patient that had had ME for about 8-9 years, was invalided by that, got Hodgkin's disease, was treated with 4 different chemotherapy regimens. After one of these 4 regimens, the patient 6-7 weeks into treatment noticed that her ME started improving. And it improved quite a lot. She got out of the wheelchair and had a social life for some months. 5 months after treatment, she started deteriorating again, gradually over some months. We did nothing with it for a couple of years. Then Øystein and I sat down and reasoned what had really happened with this patient. It was something strange. We hadn't seen anything in the literature about it. And we decided that maybe we should give her a new chance. So we sat down and reasoned what kind of drug should we give her. We couldn't give her ordinary cancer chemotherapy. That would be unethical. So we reasoned around 3 drugs, and we ended with the drug that Joe Cambridge told you about, RITUXIMAB, a drug that selectively targets B cells, B-lymphocytes, the cells in the immune system that, among other things, produce antibodies, but also have other properties in connection with interactions with other cells in the immune system. We gave her 1 single infusion. Nearly 7 weeks went on, nothing happened. We thought, okay, this was nothing. Then suddenly she calls and says that now the same thing is happening with me as when I got the cancer chemotherapy. So it's not as is said in the press that we coincidentally gave a patient with lymphoma rituximab and her ME vanished. No, it was a kind of an intellectual process before that, I must say.
You expect us to believe that, do you?
Well, it's true. Well, no one would believe us, even though we did give the good drug also to 2 other ME patients without cancer. All 3 responded, but for a duration of some months. But we thought that if anyone was to believe us, we had to do a double-blinded randomized trial. So we recruited 30 patients, 15 were given the drug, and 15 were given saline. 2 drugs, 2 infusions, 2 weeks apart, and then followed up for 12 months. And what we did see was that when we unblinded the study and saw what the patients had been given, because we didn't know and the patients didn't know what they got, we saw that the improvement of their fatigue was different, dependent on if they had got the drug or not. The red dots mark an individual with an average over a time period, and we see at once that our main endpoint was negative. That is because we constructed the study when we had seen 2 patients, and both of them were the exception. They had early responses. We decided to have the main endpoint as the fatigue score after 3 months. And you see the red and the black are the same. But if we go through the whole course of the 12 months, you see that there are more reds on top of the line, which means they're better than the black ones. But you see there are 2 black dots here in the end, and some red dots over the line. And in fact, 2 of the 15 patients were noted as responders according to the criteria given in the placebo group, while 10 of 15 of the patients in the rituximab group had response. But we knew very little still about the action of this drug. What we saw that the responses were transient. When the B cells came back in the blood, the patients gradually had recurrences. So we thought that maybe if we gave maintenance treatment, if we gave new infusions of rituximab, then the response could be of longer duration. So we had a phase 2 study. That means that all patients knew that they got rituximab. And we saw that we got good responses in the same line as we did in the other study. 62% of the patients had clinical significant responses, while 38% of the patients did not reach the level where we could call it response. We also saw that the durations were quite long. When we gave this drug up to 14 months in most patients, in 6 patients, I think it was, we gave the drug up to 24 months for different reasons. We saw that after 3 years observation, 11 of the 18 patients were still responding. But after that period, we have gradual recurrences. And that is what we really expect when we think about how this drug works. I can say that still 5 to 6 years after the trial was studied, 4 or 5 patients are still in good response without any signs of recurrence. So it seems that we can also have long-term effects of the drug, but of course being in response in the mean of about 2 years for the patient, that is not so bad when you've been where you have been. The problem for many patients is often when they have recurrence, because they've had a taste of life and going back to where they were and noticing that they're gradually getting worse, it's a terrible feeling. So the conclusion of this study was that maintenance treatment increases the duration of response. However, at the same time, we had a study for the very severe patients. We had a handful of patients. It was very difficult to get these patients to hospital to treat them in a busy oncology environment, which is our background. We're from an oncology department. And we also saw fewer responders in that group than in the more normal ME patients. So we cannot draw any conclusions concerning the effect of rituximab in the severe and very severe patients. Here's a noting of the score. 6 is a very good response concerning the fatigue. We see that the patients generally had good responses. Some of them were very good for a very long period of time. But what was my point here was that there is a lag time from when you give the drug until you see patients responding. Even these patients here at 3 months with no response, when we go further out in the curve, we see that many of them come. And I think that this tells us something about how this drug works. We interpret it as there's something in the blood of the patients that has to decrease, probably because it's not being produced anymore. There's half-life of many chemical substances. So our guess is that autoantibodies are involved. Autoantibodies, that means antibodies that don't have the beneficial effects, but they have some negative effect on the body. We also had some secondary endpoints, just to show you that. The red ones are the good responders. Looking at physical function, we see that from the baseline, they have a good response concerning physical function. The same is with vitality, bodily pain, social function, while mental health is the only one that doesn't change. And that is because the patients don't have a mental disease. It's quite simple. Well, we have to go on further, and we know then that there's a subgroup of ME patients that will probably respond to B-cell depletion. and that there's a relationship between maintenance treatment and duration of response. But of course, there are many issues to look at. Dose, for example, interval, we haven't looked properly into. But the important thing at this point was to see, can we really prove that there is a real effect in a general ME population? And to do that, you have to have a phase 3 study. And in a phase 3 study, you are to compare with the best available treatment in the other arm against the new experimental treatment. And as there is no standard drug therapy for ME, the standard arm had to be placebo. We know that placebo trials are problematic. One of the problems with a placebo trial is that when you don't have other objective measures, one of the problems there is that you get a regression toward the mean. That means the differences between the patients being given placebo and the interventional drug often are smaller. And that has to do with patients, for example, they don't know what they get. And they are so afraid of reporting a good response if they've had placebo. That's quite embarrassing. So they tend to say that the response is not quite as good. It's a psychological thing going on in their heads. Well, we managed to raise the money for the RITUXME study, which is a national 5-center study. And the null hypothesis is, of course, that placebo and rituximab are identical. And if we could find a difference, we look at the statistical chance that this happened by chance. 152 patients were the amount of patients that we were to have in the study. That has to be pre-reported. 76 patients in each group, 5 centers, started in September 2014. And we gave the same induction treatment as in our previous studies, namely 2 infusions, 2 weeks apart, 500 milligrams per square, followed by maintenance, rituximab or placebo at 3, 6, 9, and 12 months. And you note that this is not quite the same as in the maintenance study. Well, that's true. And that has several reasons. And that is, the main reason is that this study is not mainly a patient treatment. This study is to prove a concept. And we can't go on with the rituximab for 3 years. It would take much too long time to continue to do such a study. Saline with albumin was done in that way, so the nurses couldn't differentiate between the experimental drug and the placebo. And it's a truly blinded study. We don't know what we've given. The patients don't know what they've got. And we have to follow the patients until that finally included patient has been in the study for 24 months. And that happens in the end of September this year. One of the institutions got a bit later than the others, so we're a bit postponed and compared to the original timetable. And what do we have as main endpoints? Well, it's based on the patient's self-report, evaluating the changes in their fatigue. And we have a calculated fatigue score based on this. The patients tell us on a written scheme, how their different symptoms have changed from baseline every 2 weeks during the whole treatment period. Because there is a variation from day to day, we can't do it every day, but I look back the 2 last weeks, how was I compared to what I was when I went into the study? It gets problematic during the 2nd year because especially patients that alter their function, they can have problems in really remembering how it was. It gives a tendency to also underestimate the effect. We also have as a primary endpoint the number of patients in clinical response. We also have to have secondary endpoints, and they usually should try to support the primary endpoints. And we have the known quality of life SF36 and FSS schemes that are followed, as we showed in the other table. The patients are to estimate their function level every 2 weeks, also according to given written guidelines. And we also have an objective measure, because there is a relationship between how the patients feel and what the electronic registration during 7 days, night and day, tells us. In fact, we have found that what the patients tell us is very true. They are very good at telling us how they are. It has a very good correlation to the number of steps that we register. We do this before treatment and 1.5 years into treatment. We also have some sub-studies done from 1 to 3 other centers, and we'll come back to that. Of course, as a secondary endpoint, we also always have toxicity in this kind of studies. And the sub-studies are the 2-day cycling test. I won't go into that, but you know that that can be an important test for ME patients, but just a subgroup of the patients can do it, because when you do this testing, patients can get deteriorated for weeks. We have a sub-study at Haukeland, a gastrointestinal examination. We have some very interested gastroenterologists who are doing this before intervention and at 18 months. And also we look at endothelial function, and why endothelial function, which is more known from other areas of medicine, which you have in hypertension and you have in diabetes type 2, for example. Well, a group in Dundee in 2012 published a paper indicating that ME patients have problems in regulating their bloodstream, especially on the artery side. And this is caused by a lack of ability to produce the transmitter substance that the cells lying on the inside of the arteries are to produce to regulate the tone of the wall. When this is produced, you get a relaxation, you get a dilatation of the artery, and you can measure this with ultrasound if the artery goes up or down, dependent on the stimuli you use. Just wonder, yeah. Everyone says, not everyone, but many people say that rituximab is a very dangerous drug. It depends on which eyes you have. If you have an oncologist's eyes, we totally disagree. It's one of the most easy drugs to use in our practice and gives little side effects for the patients. But there are some very seldom side effects. But that's especially in very heavily treated patients which have been given a lot of other chemotherapy in addition to rituximab. So I won't go into these. The unknown thing with ME patients is if rituximab in some way can affect their disease. And what we saw in the previous study was that around every 4th patient had some deterioration in their ME condition following an infusion. We must remember that although this drug takes down the B cells, it is a kind of an immune stimulus. So it can induce something concerning the underlying disease. So this is a thing we have to look into in our study. How is the status? Well, 151 patients were evaluable. 1 withdrew of those that were recruited. And we have 2 others that have gone out of the study, 1 about a year and 1 about 18 months. So the number of patients that have completed are very high compared to most studies. The interaction between the centers has gone very well. We have had users meetings, and the data quality seems good in our hands. It comes into the center in Haukeland Hospital, and we also have external monitors that have nothing to do with the study, that just go around and look at the quality of the registration. They look at the patient journals and look how it's put into the files of the study, and they are very satisfied with that. We will be unblinding this after the last patient has finished in the end of September, after we are sure that the data is satisfactory. Then the data is locked. We have a commercial company that has the code. We get the code from the commercial company, and they also have the data. It cannot be manipulated by anyone, not by us, and the analysis will begin. And we will not tell you anything about the results before we publish this. People will be disappointed to hear that, but if we're going to get the study published, there has to be some interest in the result. If everyone knows the results, no one will publish, and that will be bad for the field. But of course, we cannot guarantee what result there is, because there are many, many phase 2 studies in the world that have shown very good promise, but when you put the patients in a phase 3 study, you can't reproduce it. Of course, when you make such a study, there are many compromises you have to do. The number of patients is one of them. If you want to be sure to have a good chance of getting a positive study, you could have more patients. But you have something called money, you have something called time, you have something called working hours. So this is a compromise. Say that some of the patients get deterioration, do not respond. Those patients can pull down the effect of responders, and that can make us get a negative trial. So as researchers, we have to be open to all kinds of results of this, and it's truly unblinded, so we don't know. We know there are responders, but we don't know if there's placebo patients responding or not. One small result, it's a baseline, just to show the kind of things we can get out of this. This is endothelial function, ability to regulate arterial vessel dimensions, flow-mediated dilatation, which is the brachial artery in the arm. Patients here, normals here. This is patients not in this trial, but in other trials. We see, look at the p-value, as a group, they have endothelial dysfunction. It doesn't mean that we can use this as a measure for each patient, but as a group, they have endothelial dysfunction. So there's something wrong with their ability to regulate the big arteries. And when we have another test for the small arteries, we see just the same thing. The patients do not dilate their arteries. They cannot adjust so quickly as normal people can. So this is a significant finding. This is nitric oxide, which is the most important factor for this ability. Now, we jump off rituximab and we go to cyclophosphamide. What in the world are we doing now? Cyclophosphamide has been around for 50 years. What are you doing? Well, again, we listened to the patients, because we had 2 patients with breast cancer, got a 3-drug regimen, which is standard in the whole country, and then they reported after starting this chemotherapy that they had improvement of their ME, marked improvement. Later, a 3rd patient also told the same story. And looking at the 3 drugs, we found out that it has to be cyclophosphamide, because it's a drug that is known to have an effect on the immune system, different from the 2 others. Cyclo is a very strange drug. It has different effects on the immune system depending on the level you give and the dosage, if you give it daily or if you give it as a pulse. So we decided to do something like what they got for their cancer chemotherapy. 3 pilot patients were given this pulse treatment every 4 weeks for 6 times, and 2 of the 3 had a good effect. A 3rd had no clinical response, but there were indications that something also happened to her ME. And this ended up in applying for a phase 2 study, 40 patients. We deliberately recruited 15 patients that had been in previous studies. Some had had rituximab with response, and some had not response, and some had, all of them had had rituximab, but all of them had recurrence of the disease. 25 patients had no previous drug treatment. It's a 2-center study, Oslo and the Norwegian Radium Hospital, Oslo University Hospital. Here, the intention was to get a bit sicker patients than in the RITUXME study. And one of the reasons for this is that cyclophosphamide is much easier to give outside of a more specialized unit. You don't have to have 4 to 6 hours infusion. You don't have to have specialized nurses following the patients. It can be done in an hour in a small community hospital. And we have in this study the same endpoints as in the RITUXME study. And we're finished with this study in July, and we will have the analysis of the data going on during the autumn. What we can say already and that's interesting, is that we do truly see responses also in this group. We also, I can say that there are patients responding to rituximab with recurrence that say that they have responded just as well to cyclo. So that's interesting. On the negative effect, there's more subjective toxicity than in cancer patients. There are more nausea, vomiting, and there's more accentuation of the ME symptoms while they're getting the treatment than we usually have seen in rituximab. Very little hematotoxicity. We can hardly see that there's any change in the white blood cells. Problem is for 35, 40-year-olds, women, that you can get menopause induced earlier than the patient naturally would have had. So this is a thing that you have to consider. But the interesting biological aspect here is that you are now giving a more broadly acting immune drug and nevertheless get a response. And that is interesting. Just now we're starting up a new study for 5 patients. It's going to be expanded to 20 if it works. And we're treating then severe and very severe patients and see if this can work in practice. What we then are doing is that we increase the interval to 6 weeks instead of 4, both to see if we have the same response and if it's better tolerated. One of the great advantages of doing clinical studies is that you have a patient population that is very well characterized, and you can accumulate a big biobank. And that is what we have used when we hear the next presentation. Dr. Haukeland from our institution will tell us something of what Øystein should have said. And we see that here we have a lot of samples. We have already a good international cooperation where we use the expertise of other groups, where they can help us to understand, and we together can understand the total pathomechanisms in this disease in a better way. Of course, the interesting thing here is also we have a manipulation that will give responses in some patients. We can see what happens to the same markers when the patients respond. What happens to patients, for example, who have spontaneous bettering during treatment? Does the same things happen with those markers as in patients that respond to rituximab? Just a small taste of this. In our metabolomic study that we published around New Year, we took serum from 6 quite sick ME patients and let muscle cells grow exposed to serum, and we measured what happened to their metabolism, especially oxygen consumption rate and extracellular acidification rate. And we compared this to 6 healthy controls, and what we did see was there was a clear difference in what happened. We can see that there's something in the serum of the patients with ME that makes normal cells behave differently. And that's very important. Before this publication, I had a public appearance, and I said something like ME-CFS is in the blood and not between the ears. And I think that is proven. Well, it's no big knowledge for the patients. They've known it all the way. It's health professionals and the public that have not known this. And that's our message to tell them. This is the way it is. Okay, so our present hypothesis is that ME is an immune system disease. We will hear that we have good indications that it's a mitochondrial defect, not only maybe, but that's a part of it. And that lack of ATP in maybe multiple cells, not only the peripheral blood cells that we're looking at. I would suggest that many types of cells in the body, maybe all cells that have mitochondria are affected. And we get widespread symptoms. And I think that as a reaction to this, we also see compensation mechanisms from the body. So what we're looking at, we can't really say, is it induced by the disease or is it some kind of compensatory mechanism going on? And that, I would say that if we look just very narrowly at some specific factor, we can think this is the answer. But it doesn't have to be. It can be very, very far away from where the action really is going on. But I would say that what we see with rituximab and now with cyclophosphamide is that when patients start improving, all symptoms usually improve at the same time. Not always to the same degree, but at the same time. And that indicates that there is some key event that can be triggered and that will make the patients improve. Okay, what about the future? Well, it's dependent on the outcome of the RITUXME study. If the study shows benefit, then we have an ethical obligation to the group that has been given placebo. They know that when they go into the study, they have a 50% chance of not getting an active drug. And we have an ethical approval to give them the opportunity to come into a new study where they know they will be given rituximab. So there will be other things to be answered in a new study, if we can get the money, of course. They are costly trials. And we're going to try to keep our multi-center organization going. So we're trying to pay them this autumn to keep them interested. And we'll see after New Year. We will, under all circumstances, be doing more studies on cyclophosphamide. Here's our group. In fact, there are 4 people that get paid, mostly through the RITUXME study. All the others have positions in the oncology department and are working just small percentages of their time. You see Øystein here. You'll also see another picture of him, I think, afterwards. And this is Carl Tronstad. He's the professor of biochemistry, who's the main author. No, he's the senior author of the metabolomics study. Here are all the cooperating doctors and other people in other institutions, and all. We are. The interest in ME has increased quite a lot since we started in 2008 in our institution. And I hope also that we can continue contributing in this field so that we push the treatment options for patients further in the positive direction. I think that a clue to the acknowledgement of the field is to find out more of the pathomechanisms. So all the work going around in the world is extremely important to get there. And I'm very optimistic for the future. That is my feeling. Thank you.
Can I say that everybody I've talked to who's known you since 2008, including myself, I've been absolutely impressed by the way you've handled this science, how careful you've been, how no big claims have been made until everything's been assured. You seem to respect and understand how important it is to many people, and I congratulate you for that. And could you take our congratulations to Øystein too and say we hope to see him soon too. I don't know how you sleep at nights when such data is lying in an envelope. I don't think I could. I think I'd get the scissors out and get into it. But please be careful, carry it on, and we look forward to that day when you can come back and say, well, we've got something positive here, but, because you will, I know, there's always something else you ask about, but the patients knowing that something like this is going on and been waiting for it have a lot to say thanks for, and they will. So well done, and to your group too. It just encourages everybody else, and I hope many of the people listening here are encouraged to carry on the research and to not give up at the first hurdle when there's something funny happening that you don't understand. So thank you. I just... And give our regards to Øystein, and we're sorry he's not here. Tell him to keep away from ladders till he's, a bit older.